Um, so today I'll be introducing scDEED, which, as Jessica said, was a statistical method for detecting dubious 2D single-cell embeddings. So in analysis, many times we move from the high dimension to the low dimension, like moving from the gene expression space into something 2D like t-SNEAnd or UMAP so we can visualize the data. But this conversion isn't free, it comes at the cost of distortion. A more everyday example might be a three-dimensional globe and a 2D map. So even though the world is three-dimensional, a lot of the times we look at two-dimensional visualizations because this is a little bit easier to see on our laptops or on a sheet paper. But there are some known distortions, like here if we look at Greenland and South America, it looks like they're about the same size. But actually, South America is 9 times larger than Greenland. So there are things in the visualization that are not quite so trustworthy based off what we can see. And so a related question then becomes, what are the distortions in single cell data? And this is a much harder question to answer because we don't have a physical analog like we do for the globe. And then there's the additional complication that popular methods like t-SNEAnd and UMAP are very sensitive to hyperparameter selection. So here's a toy data example where we applied t-SNEAnd at 3 different perplexities. And each of the visualizations are different. The relative location of the clusters is different, like here the blue and the green. And then also some of the clusters have been split, like this green cluster here. So now we have a new question as well, which is what hyperparameter should I use? So some current practices, sometimes people just use the default parameters. Usually this turns out to be like perplexity 30 or 40 for t-SNEAnd. There are also some general guidelines, like the number of cells divided by 100. And then there can be visual selection. So some researchers will try a variety of hyperparameters and then select from the output visualizations. The problem with that is that it's not very replicable and really depends on what the researcher expects to see. And then for these first 3 options too, we also have the additional issue that we're not really addressing where the distortions are. So there may be things that are not fully replicated in the 2D visualization, so it doesn't quite represent the high dimension, but the first 3 options are not going to help us locate what might have been distorted. The last option is optimization of some sort of loss function. And the goal here would be to try to categorize the cells and their embeddings to identify the distortions or to help optimize the hyperparameters. And there are some existing methods, which I will discuss later, but this is also what scDEED is going to try to do. So I'd like to introduce scDEED, a method whose purpose is to identify cells whose 2D embedding is visually misleading or dubious. And by dubious, I mean that the cell's location in the 2D embedding does not faithfully represent where it should be based off the higher dimension or pre-embedding space. Based off these dubious cells, we can then suggest optimized hyperparameters by minimizing the number of dubious cell embeddings. So the main idea behind scDEED is that the 2D embedding space should look like the pre-embedding space. And we evaluate this through neighbors. So in the 2-dimensional space, we calculate a distance between a cell and its nearest neighbors, with the nearest neighbors defined either by the 2D embedding space or the pre-embedding space. So oftentimes this might be the PCA space, which is k-dimensional, with k input by the user. We will then compare these distance vectors to create a reliability score, which is really just the correlation of the 2 distance vectors. If the agreement is good, like for this purple cell, where the pre-embedding neighbors look very much like the 2D embedding neighbors, then the correlation is high. But for other cells, the pre-embedding and post-embedding neighbors might be very different. Like here, we can see that the pre-embedding neighbors span 2 clusters, whereas in the embedding itself, it looks like the orange cell is just part of this cluster. So this orange cells, their position is more misleading, and so this correlation will be lower. and we'll get a lower reliability score. We can further evaluate high and low by looking at permuted data. So permutation will remove prior cell-to-cell relationships and disrupt all the embeddings. So any correlation we see is just due to random chance. By calculating reliability scores on all of this permuted data, we obtain the null distribution. We can then threshold on the null distribution to classify the cells as either dubious or trustworthy, with dubious again meaning that the cell's neighbors in the 2D embedding do not faithfully represent the pre-embedding space. So one thing I also want to point out is that although our package is currently designed for t-SNEand and UMAP specifically, actually this framework can be applied to any dimension reduction method. So really this can be used to compare dimension reductions to each other, not only optimize hyperparameters. For my first example, I'd like to talk about the Hydra dataset. This was a single-cell RNA sequencing dataset of the Hydra polyp. On the left, we have a reproduction of the study's original figure, which was done at perplexity 4D. We applied scDEED and found the optimized perplexity to be 230. To get a better idea of the differences in these visualizations, we looked at the dubious cells. So at perplexity 230, there are very few dubious embeddings, and they're mostly just on the periphery of clusters. But in the original embeddings, we have entire clusters that have been marked dubious, suggesting that this cluster location is not reliable. We took a look at some of the dubious embeddings. So here specifically is the neuron EC1 cell type. In the original embeddings, the neuron EC1 cell type appears in two clusters, one that was marked dubious by scDEED and one that was marked trustworthy. But actually, if we look at the gene expression of the two clusters, we find that their gene expression is quite similar. And also, the authors did annotate it as a single cell type. So the fact that this cell type has been split into two is a little bit misleading. We can also look at the orange stem cell cluster, which is between our two neuron EC1 parts. Because of the proximity, we might expect that all three of these clusters should have similar gene expression. But actually, the orange stem cell cluster has totally different gene expression compared to the neuron EC1 cells. So we have two inconsistencies. The first is that the cell types are separated, and the second is that different gene expression is actually very close in the visualization. Under the optimized perplexity of 230, we actually find that both of these have been resolved. So the neuron EC1 embeddings are now unified in a single cluster. And the fact that they're unified here also really supports that this apparent split is not real and biologically meaningful, but just an artifact of dimension reduction method. Because if we change the perplexity, it goes away. And also we find that the orange stem cell cluster is now very far from the neuron EC1 embeddings. It's a little bit more intuitive because we have that distance in the visualization corresponds to differences in gene expression. We also took a look at the sensitivity of the hyperparameter. So on the top, we have, it's actually the same graph on the top, but the number of dubious cell embeddings found at different perplexities by scDEED. And so this is the original perplexity of 4D. And there are many ways to pick the critical point here. So one is you could use just the global minimum, which is what we do with scDEED. But there are other methods to pick the critical point, like the needle method, which looks for the elbow point in a plot here. And that finds the optimal perplexity to be 170. But actually, if you look at perplexity 170 and you look at perplexity 230, these really look very much the same. And if you look for the neuron EC1 cell type, which isn't very easy to see, but it is in this cluster here, and our EP stem cells are also still up here. So many of the things that we noted about the optimized perplexity are still true at 170. And so one thing we found was that it's not that there is one magic perplexity number, but rather there's a range of perplexities at which you can obtain an optimized visualization, and scDEED can help find that optimal perplexity range. So far, the benefits of scDEED include that the dubious cells can highlight spurious clusters, and also that optimization can improve the cluster distance and gene expression relationship, where distances between clusters should correspond to differences in gene expression. For my second example, I'd like to talk about RNA velocity. So as a brief review, to make proteins, we start with DNA, which is then transcribed into pre-mRNA. This pre-mRNA has to undergo RNA splicing before it can be used to make proteins. But RNA sequencing will actually pick up pre-mRNA, which is unspliced, and proper mRNA, which is spliced. So the idea with RNA velocity is that we can use the amounts of pre-mRNA to estimate where the cell is going to go. So we're using the conversion rate to get a little bit of a time estimate of how the gene expression is going to change based off the amount of pre-mRNA and unspliced mRNA that we see. So these are some figures from the Lamano velocity paper. Here's just again, the transcription going to unspliced, then spliced. So what RNA velocity will predict is a cell-specific gene velocity vector. So each cell gets a specific velocity vector, which is very high dimensional. But when it comes to visualization, especially when you're using something like t-SNEAnd or UMAP, as most people do, we can't visualize this very high dimensional velocity vector. So what's actually plotted here is a local group velocity. So around each of these arrows, we have a kind of box. And what this arrow is representing is the local group velocity for the cells in that box around it. So most RNA-seq analysis happens in the high dimensional space, but RNA velocity is a special case because it is highly dependent on the visualization. This embedding will determine which cells are included in that box for the local group velocity. So here we used the mouse dentate gyrus dataset, which is the dataset used in the tutorials for RNA velocity. The original one was done at perplexity 30. We applied scDEED and found the optimized perplexity to be 450. At first glance, these visualizations look very much the same. Maybe the relative location of the clusters around here have changed, but overall, pretty similar. However, the RNA velocity is totally different. So in the original visualization, we can see this general trajectory from these NIPC cells into the neurosubtypes and then the immature granules. But if we look at the optimized perplexity, we see a much stronger visualization where the arrows have been much exaggerated. And what this means is that the little boxes around this arrow, that local group velocity is in a much better agreement, which is how we're able to get a more exaggerated and more clear trajectory. But I also want to point out that these mature granules, which are fully differentiated, and so we might not expect to see any velocities there because they're kind of done. And we don't see an exaggeration of velocities here. So in the scDEED optimized visualization, we do see an exaggeration of trajectories that we should see, but we're not seeing exaggeration of trajectories everywhere like these mature granules. We don't see exaggeration there, and we don't expect to. For my last example, I would like to talk about some of the other methods that work to do something similar. So this is Embedder, a method that also works to classify cells as embedded or noisy. This would be our version of trustworthy and dubious. And the way Embedder works is by comparing the cells pre and post embedding, very much sort of like scDEED. So what they do is they take the data and turn it into affinity matrices. And they do this using a Gaussian kernel for the high dimension and the student's t-distribution for the low dimension. And say how similar cells are to each other. Then the student's t-distribution to evaluate the low dimensional space, and then compare this through the KL divergence. So a very similar process to t-SNEAand. The only difference here is in the null data distribution, where they not quite permute data, but will randomly sample gene values for each of the cells, and then apply the same matrix to get a null distribution of EES scores, which they can then threshold on. And in this case, actually, small means good. Small means the high and low dimensions have a very good KL, a small KL divergence, so they're very similar, and that would be well embedded. And larger values would be a more noisy embedding, where these two affinities are not quite so in agreement. So overall, it's a very similar process as a scDEED, but the major downside is that this is based off the t-SNEand loss function. So some consequences of being based off the t-SNEand loss function. First is that as we increase perplexity, the number of dubious embeddings goes down. And so this always happens. This is known to happen with t-SNEand, which is why you can't optimize t-SNEand perplexity using the KL divergence, because increasing perplexity will get you a smaller KL divergence. We also have that t-SNEand and UMAP can't be compared, because actually Embedder is optimizing the same thing as t-SNEand, and so t-SNEand will always get a smaller score than UMAP. And we see that same thing here when you look at the same k-effective values. K-effective is something designed by Embedder to estimate the number of cells that are used in the neighborhood calculations. And they define k-effective so that we can compare t-SNEand and UMAP. And so here, when we use the same k-effective value, actually UMAP always has more dubious embeddings compared to t-SNEand. But we also have that scDEED will outperform Embedder. So here's the mirror data set, which was used in the Embedder paper, and Embedder found the optimized perplexity to be 1,000, whereas scDEED found the optimized perplexity to be 100. So these visualizations, I think the biggest difference is in the location of the KLS and granulocytes. In the scDEED optimized version, they are almost touching, and in the Embedder optimized version, they are very far apart. To examine whether or not this was valid, this proximity, we looked at a KLS cell in this sort of shared region here, and then looked at the 15 nearest neighbors based off the PCA space. And actually, the 15 nearest neighbors for this red KLS cell are in both KLS and granulocytes. So this proximity in clusters is warranted because it does accurately reflect the cell's nearest neighbors. Whereas the Embedder optimized version would have lost that information because the granulocytes would have been quite far. So in this one, scDEED did better preserve the neighbors. So some of our conclusions for scDEED, one is that the use of dubious cells can help highlight distortions. So just as there are distortions in the continents when we move from the globe to the 2D visualization, there can be distortions in our single cell data. And by evaluating the dubious cells, we can find out which parts of the visualization may not be so trustworthy. This can also help us identify spurious clusters, which are not biologically meaningful, but just an artifact of the dimension reduction method. Using these dubious cells, we can also help optimize hyperparameters, which can result in less distortion in our visualization and better agreement among the cells. And this is evident through the RNA velocity visualizations. And then scDEED is also very flexible because its loss function is unrelated to dimension reduction methods and their loss functions. scDEED can be used to compare dimension reduction methods to each other and can also be used with any dimension reduction method. So much more flexible. And so finally, I'd like to acknowledge the contributions of Lucy and Jessica, the co-authors, t-SNEand Liu for all of his work writing the scDEED package, and the other JSB lab members for their support. And as well as funding sources like the NSF Mentorship Grant and the UCLA Department of Statistics and Data Science. And that's the end of my presentation. If anybody has any questions, I'd be happy to take them.